Welcome back, everyone. In today's lecture, we are going to talk about the control of renin release, a critical mechanism that regulates blood pressure and fluid balance in our body. By the end, you will not only understand these mechanisms but also be able to tackle MCQs confidently. So, renin is an enzyme secreted by juxtaglomerular cells in the kidney. Its main job is to kickstart the renin-angiotensin system by converting angiotensinogen into angiotensin 1. Here is the important part. First is active renin is released when the body needs it, like during low blood pressure or sodium levels. And second one is prorenin is released continuously even when it's not immediately needed. This can make up to 80 to 90% of total renin in the bloodstream. Think of prorenin as a reserve and active renin as the player on the field. This distinction often comes up in the MCQs. Now, the first factor is about macula densa mechanism. So, the macula densa is a group of cells that located near the distal tubule of nephron, and these cells act like sensors for sodium chloride concentration in the fluid flowing through our tubule. If there is high sodium concentration or NaCl concentration, the macula densa signals the kidney to reduce the renin release. If there is low sodium chloride concentration, it stimulates renin release to increase the sodium reabsorption and restore the balance. Now, the question is, how does it send this message? Obviously, through the chemical mediators. Now, which kind of chemical mediators? So, the first is prostaglandin E2 or PGE2 and the nitric oxide or NO, which increases the renin release. And the second one is adenosine, which actually reduces renin release. Imagine a car fuel gauge. If it senses low fuel, like low sodium, it signals the driver or kidney to refuel, means release the renin. If the tank is full or high sodium, no action is needed, obviously. Now, the renal baroreceptors. So, renal baroreceptors detects changes in the renal artery pressure. This mechanism works inversely. Like if there is high pressure, renin release decreases. And if there is low pressure, renin release increases. Now the question is why? So when the blood pressure increases, it stretches the juxtaglomerular cells. This stretch causes calcium ions to enter the cells. Paradoxically, higher the calcium levels inhibit the renin release. Think of it as a balloon. If it's stretched too much, means high pressure, you stop adding air, means renin. If it's deflated, means low pressure, you add more air, right? Next up is the sympathetic nervous system, which comes into play during the stress or injury or is if there is any low blood volume. So it releases norepinephrine, which directly stimulates renin release through beta 1 adrenergic receptors on the juxtaglomerular cells. This system is particularly important in emergencies like when you are dehydrated or bleeding to quickly restore blood pressure and fluid balance. Picture your body like a fire alarm system. When there is a drop in pressure, the sympathetic nerves sound the alarm by releasing the norepinephrine to stimulate renin release. Now, the fourth factor is angiotensin 2 feedback. So angiotensin 2 provides a negative feedback loop to regulate the renin release. When angiotensin 2 levels are too high, it tells the kidney to reduce the renin secretion. It does this in two ways: by rising blood pressure, which is sensed by renal baroreceptor, by directly acting on juxtaglomerular cells through an increase in the intracellular calcium, which actually inhibits the renin release. Think of angiotensin 2 as the brakes on a car. When the system is working too fast, it slows down the things. Now let's go inside the juxtaglomerular cells to understand how renin release is controlled at the molecular level. Three main intracellular messengers play critical roles here. First messenger is cyclic AMP or cyclic adenosine monophosphate. Cyclic AMP is like the on switch for renin release. When the cyclic AMP levels rises, renin release increases. This can happen through activation of the enzyme adenylyl cyclase, which actually produces cyclic AMP. Alternatively, drugs like phosphodiesterase inhibitors prevent the cyclic AMP breakdown, leading to higher levels and more renin secretion. Imagine cyclic AMP as a delivery app. The higher its activity, the more active renin is delivered to the bloodstream to handle the blood pressure issues. Now, the second chemical mediators or the chemical messengers is cyclic GMP or cyclic guanosine monophosphate. CGMP plays an indirect role by modulating cyclic AMP. High levels of CGMP or cyclic GMP obviously tends to produce tends to reduce cyclic AMP levels, which leading to less renin release. This is why substances like nitric oxide and natriuretic peptides, which increases cyclic GMP, can indirectly inhibit the renin. So if cyclic AMP is the gas pedal, cyclic GMP acts like a safety system that keeps the car from the accelerating out of control. Now the third one is calcium. So calcium acts as a brake system for renin release. When calcium levels inside the juxtaglomerular cells increases, renin release is inhibited. High calcium can come from the influx of calcium 2 plus into the cells or its release from the intracellular stores. Now the question is, why is this interesting? Unlike most systems where calcium triggers secretion, here it does the opposite. The paradoxical effect is because high calcium levels stabilizes the juxtaglomerular cells and prevent renin from being secreted. Think of a hosepipe with a valve. If calcium builds up, it tightens the valve and reduces water or renin flow. Now, the sixth factor is pharmacologic modulation of renin. So, pharmacology adds another layer of complexity to renin control. Certain drugs can increase or decrease renin release depending on their mechanism of action. Let's break it down into two categories: drugs that increase the renin release and drugs that decreases the renin release. So first I will discuss about drugs that increases the renin release. So these drugs actually stimulate the body to produce more renin, either by lowering the blood pressure or directly acting on the kidney. So these kind of drugs are vasodilators like hydralazine, minoxidil, and nitroprusside, which relaxes the blood vessels and causing the blood pressure to drop. In response, the kidney increases renin release to restore the blood pressure. Beta adrenergic agonist medications like isoproterenol stimulate beta 1 adrenergic receptors on the juxtaglomerular cells, directly increasing the renin release. Other drugs are phosphodiesterase inhibitors, drugs like theophylline and milrinone. So this kind of drugs increases cyclic AMP levels by preventing its breakdown. So more the cyclic AMP equal to more the renin release. And diuretics like furosemide obviously reduces the sodium levels and blood volume and activating the macula densa and the baroreceptor mechanisms to stimulate the renin. Now the drugs that decreases the renin release. So these drugs suppresses the renin release often to manage the blood pressure or heart conditions. Among these, ACE inhibitors or angiotensin converting enzyme inhibitors, drugs like enalapril, which blocks the conversion of angiotensin 1 to angiotensin 2. This interrupts the negative feedback mechanism or loop and reducing the stimulus for renin release. ARBs like angiotensin receptor blockers, medications like losartan, block the angiotensin 2 receptors and preventing its inhibitory effect on renin release. This also disrupts the feedback loop. Beta adrenergic blockers, drugs like propranolol, block the beta 1 adrenergic receptors and directly reducing the renin release by preventing the sympathetic stimulation of the juxtaglomerular cells. So, renin release is a beautifully coordinated process involving multiple factors and pathways. Understanding the stimulators and the inhibitors along with the roles of drugs help us not only grasp the physiological concept but also answer tough MCQs with confidence. If you enjoyed this lecture, give it a thumbs up, share it with your friends, and don't forget to subscribe to the channel for more pharmacology related content. See you in the next video where we will explore more high yield topics.